cell division, and the cell cycle. So our objectives for this unit, it's going to be a combination between the cell cycle and genetics. You're going to be able to identify and create models of mitosis and meiosis. You're going to figure out what cell specialization is. You are going to provide some reasonings behind genetic diversity. Why is it an advantage for populations? You're going to compare and contrast the two different processes. You're going to explain Mendelian's law of heredity. You're going to identify traits. You're going to use a Punnett square in order to figure out those traits, and you're also going to evaluate a karyotype based off of different determining gender versus the genetic health. So when we're talking about DNA, we're actually looking at a particular segment. The segment is called a gene. Now, one DNA molecule contains thousands upon thousands of genes. Until they reach or wrap into, okay, they're going to wrap around histones. And they eventually become chromosomes. So again, you've got your double helix. They're going to wrap around a protein called histones. That wraps into itself, called a supercoil, even more into a supercoil, which then eventually it wraps into or condenses chromosome. Now, there's two parts to a chromosome. Now, each of the parts are called chromatids. Now, one half of a chromosome is called a chromatin. Now, there's two of them, and therefore we call them sisters. And the middle, now you get one from mom and one from dad. So again, each chromosome is made of one part, which is one sister chromatid versus another sister chromatid. Together they form a chromosome. Called a centromere. There's two different types of chromosomes. You can have diploid chromosomes. That means you have two homologous pairs, or 2N. This is your body cells. Okay. In humans, we have 46 chromosomes. To a diploid or a diploid zygote, because again, 23 largest to the smallest. Now this is an actual picture. Okay, so you'll notice that each pair is a homologous pair, right? You get one from mom and one from dad. Until you have 22 pairs, and our last pair, which is 23, this is referred to as your sex chromosomes. Now pair 1 through 22 are autosomes. That means that they are per your body. Okay, so coding for your hair, your eye color, your brain, your kidneys. Okay, it's going to code for all of that. Now the last pair, X and Y, that's going to code for, and that's going to determine your sex or your gender. Are you male or female? And it's also going to code for those particular characteristics that are associated with those sex chromosomes. So for this person here, if we look at pair 23, it is an X and a Y. Now, an X and a Y means that it is this person, this individual, biologically Now, some abnormalities that you might see. That's why we look at when somebody says, oh, I'm getting genetically tested. They are going to look at, the geneticists are going to look at and create an Now, again, we talked about the sex chromosome. Now let's talk about cell division. Okay? So cell division, we are talking about synamic these are your sex cells. So there's 4 different reasons why a cell needs to divide. Okay, it needs to grow. It was damaged and it needs membrane. So there's 2 parts. There's interphase that has G1 So if we look, which out of those 3 phases are the largest, or the longest? Interphase, all the blue, right? G1, which is cell. Now, remember, the cell cycle has 2 divisions, but mitosis is part of that, okay, and it actually has 4 stages. So in order to... Now, let's look at interphase. Phase, this is where the majority of the cell is going to be part of the cell cycle. Okay, so this is, we also refer to it as the resting stage because it's not really dividing. Now, G1. Okay, this is when it occurs after Now again, alright, so this is a karyotype. Okay, so karyotypes are used for determining chromosomal disorders. So in us as humans, we have 46 chromosomes in each of our body cells. Now, we get 23 of those chromosomes from our mother and 23 chromosomes from our father. So each of these pairs, we have 23 pairs. Each of these pairs, you get 1 from mom and 1 from dad. Okay? Now, your pair 23, that's really special, okay? They are called your sex chromosomes. So, for this example, this person has a X and a Y. Now, in order to be, if you receive X and a Y, you're referred to as a male. If you receive two X's, then you're now, interphase. Okay, so interphase is our very, very first phase of mitosis. Okay, so remember, mitosis, we're making body cells. Now, if you so mitosis, okay, mitosis is the part of the cell cycle where the nucleus actually physically divides. Okay, so in order for this to happen, you're going to make an exact replica or an exact copy of the original DNA or the parent DNA. Okay, so it's like me going to the Xerox machine and making a copy of a worksheet. You're just going to make an exact replica. Okay, it's not exact same. Now, we just touched a little bit on interphase, okay? So interphase is the first step of the cell cycle. Now, mitosis is its own section of the cell cycle, okay? So you have prophase. Prophase is the first step of mitosis, okay? So it is it's underneath that umbrella of the cell cycle, but mitosis is only 4 steps, okay? So prophase is the first and longest phase of mitosis. This is where you can actually see those physical chromosomes, those X's, okay? So before in interphase, like that picture before, you couldn't see a physical X. They just looks like worms or a bowl of spaghetti, okay? Those centrioles, those little Twizzler bites, they're going to start moving to opposite sides, okay? The nucleolus and the nuclear membrane, it goes bye-bye. You cannot see that anymore. The spindle fibers basically are going to form and they're going to be attached to those centrioles. So they're going to start moving to opposite sides. Okay, so these tubules or these microtubules actually help the chromosomes separate when we go down through our cell cycle or through the steps of mitosis. Okay, and you see a picture here. So again, main takeaway, the chromosomes, you can see the chromosomes, the nuclear envelope is disappearing. Okay, those centrioles are going to move. So that's the takeaway. You want to take away prophase, first phase, longest phase. You can see those X's, the nucleus. Now, during prophase, okay, so these chromosomes actually are joined together. So each chromosome is made out of one sister chromatid, okay? So a chromosome has one side is a one sister chromatid. The other side of the chromosome that makes that other X or that other line, that is going to be the other sister chromatid. Now, again, you have two sisters in order to form that X or that chromosome. Now, in the middle where they're actually being connected, that's called the centromere. You can think about it, center is. Now metaphase is really easy to remember. You can remember it. Meta means middle, okay? Meta, middle. Now these chromosomes are going to line up in the middle or the equator, if you will, or the central zone of the cell. Okay, so again, they're attached, these spindle fibers are attached, they attach themselves to the centromere or the center part of the chromosome. Now, the centromeres, or excuse me, your centrioles, they are also attached to those spindle fibers, and they're the ones that are actually going to be pulling these chromatin. So again, this is where you have that centromere. It's going to connect itself. Now, the spindle fibers are basically like fishing line, okay? And it's going to pull either, or play like tug of war. It's going to pull them into a direction, either to the northern hemisphere or the southern hemisphere, or excuse me, the western or the eastern hemisphere of the cell. If you is. Now, Anaphase is not very nice, okay? Anna's not a very nice lady, okay? She likes to rip people apart, okay? So you can remember it as the chromatids are going to be separated. They're going to be split, okay? Those chromosomes are no longer going to be chromosomes. They're going to be split apart, okay? So this is where we actually start forming daughter chromosomes. Now, telophase. Telophase is the very last step of mitosis, okay? This is where the nuclear envelope or membrane is going to start slowly reappearing, okay? So this is where DNA is going to start uncoiling, okay, back into those chromosomes. Individual cells. Okay, so again, mitosis. Prophase, metaphase, anaphase, telophase. Okay, those are the 4 steps for mitosis. So, parasitic, we talked a little bit about that cleavage furrow. Okay, so in animal cells, it's called a cleavage furrow. Basically, it's daughter cells. Plants are a little bit different, okay, because plants actually have a cell wall, okay, on the very outside compared to their cell membrane. Okay, so the cell wall is actually going to, the cytoplasm is going to divide into a okay, that cell plate is going to eventually be that cell wall. Now, click on the video, take a couple minutes, check it out. Alright, so in your graphic organizers, you have this chart. I want you to take several minutes, and I want you to go ahead and copy this information down. Okay, so these are the major events of interphase. So interphase, you have G1, S, G2. Okay, so what are the major events? What are things occurring during G1? Well, it's going to grow rapidly, okay? Because again, this is normal routine. This is its daily function. This is doing its own thing, okay? So the cell is going to start maturing. It's going to increase in size, okay? Because again, it's making new organelles, okay? And it's going to make new cytoplasm. So it's getting itself ready, Okay, so S phase means to synthesize. Okay, so synthesize means to make. Now, what exactly is it making? It's going to make DNA replicate. So again, it's going to go through DNA replication, which we're going to talk about later. But DNA is going to make a copy of itself. Now, why does it need to make a copy of itself if we're getting ready to make a copy? Well, because DNA replication goes through its own set of copying instructions and copying techniques, okay, compared to the actual cell itself. So again, we're trying to make it a little bit more efficient in making compared to doing it all together, okay? So the chromosomes are going to start to coil. You're going to start seeing them condense, okay, very, very, very slowly. Now G1, this is where it's just going to prep itself for cell division. Okay, so it's going to get prepared. Kind of think about it like you're getting hyped to go out on the field or get ready for your basketball game. Okay, so you're in the locker room and you're getting pumped and you're getting ready. That's basically what G1 is going to do. Okay, it's going to get itself started. Alright, so just like we said before, okay, we're going to go ahead and copy down the mitosis or the M phase. Okay, so we've got 4 phases: prophase, metaphase, anaphase, telophase. Okay, so again, prophase is the very first, and it's the longest phase of mitosis. This is where you can actually physically see the chromosomes. Okay, they're starting to be able to be visible. Now, the centrioles and the spindle fibers are going to start forming, and the centrioles are going to start separating and moving to the opposite sides of the cell. Now, the last step is where the nucleolus and nuclear membrane, it disappears, okay? It's going to be vanished. Now, meta means middle. The chromosomes are going to move and align in the middle of the cell. Remember, Anna's not a very nice lady, okay? So the daughter chromosomes are going to be pulled towards the opposite ends of the cell, okay, by those spindle fibers. Remember, it's kind of like tug of war. They're going to be pulling to their side of the cell. Now, telophase, this is where the DNA is going to start uncoiling itself and starting to turn into the chromatin format. And this is where you're also going to start seeing your nucleolus become visible again. So again, M-phase, prophase, metaphase, anaphase, telophase. Those are the 4 steps for mitosis, okay? Now, interphase and cytokinesis are not part of mitosis. It's only part of cell cycle, okay? So cytokinesis, this is, again, mitosis is where the nucleus is going to divide. Cytokinesis is where the cytoplasm divides, okay? So cyto, cell, okay? That's what cyto means. Now, this is where you're actually going to see 2 identical daughter cells, okay? And it's an exact copy. Remember, an exact body. So, we've already talked a little bit about, again, the cell cycle. So, the cell cycle, okay, is when we talk about interphase, prophase, metaphase, anaphase, telophase, and then therefore cytokinesis. Okay, so which one is the longest phase of the cell cycle? Interphase. Very good. Okay, can you name the 3 phases of interphase? G1, S, G2. Excellent. Okay, what are the 4 steps or 4 phases of mitosis? Prophase, meta, ana, tele. And what is cytokinesis? What is it dividing? Nucleus or cytoplasm? Alright, so when we talk about cell division, okay, so who does cell division? Okay, what kind of organism? Believe it or not. Now, there's 2 different types of reproduction. There's asexual, And then there's sexual. Okay, so asexual means you don't need a partner. You can do it all by yourself. Okay, so pretty much you're going to make an exact copy. Okay, so again, do we make an exact copy when we talk about mitosis? Absolutely. But in this case, are we making an exact copy of a body cell, or are we making an exact copy of the actual organism itself? Very good. Okay, we're actually making an exact copy of the organism itself. Okay, so if you're single cellular, you're only one cell, you need to go through mitosis because you're just making an exact copy of your whole self. Okay, so prokaryotic. Okay, anything that has any type of cell or organism that does not have a nucleus, it's going to reproduce asexually. Okay, there are a couple eukaryotic organisms. Okay, again, eukaryotic means like us, we have a nucleus. That's like sponges. Sponges actually have the ability to reproduce asexually as well if they can't find an actual partner themselves. Now, what's so important about sexual reproduction? Okay, so again, the offspring, okay, like us, we had to come from two different types of sex cells, okay? One from mom, one from dad, right? Two parents. That's going to fuse into one, okay? If you just have a sperm and you just have an egg in two separate locations, are they going to turn into anything? Nope, they're not going to make an offspring, okay? Because you have to have the genetic information from both parents, okay? So plants, animals, some single cellular organisms, not many, they do reproduce sexually, okay? But this form of sexual reproduction, we do not make clones, okay? So you cannot take an egg, you cannot take a sperm and then make an exact replica. Nope, because you don't look identical to your parents. You might have similar traits, but you are not an exact replica. You are not an exact copy, okay? So, again, asexual, they go through mitosis because they're making an exact copy of themselves. Sexual reproduction, we actually have to go through meiosis, which is a different format when it comes to our cell division. Now, major difference between mitosis is body cells, and meiosis, which is sex cells, okay? So meiosis is very, very similar. But at meiosis, there's 4 gametes, or 4 sex cells. So you have eggs or sperm. Now, how many eggs do you get per month? You only get 1, okay? So each time you go through meiosis, as females, okay, we get 1 egg. Well, Ms. Gwen, you said 4 at a time. Well, where's the rest of them? Okay, so you get 1 egg, and those last 3 are called polar bodies. These polar bodies are basically trash, okay? So they leave your body, okay, at the end of your menstrual cycle, okay? So these are polar bodies. These are trash. So you get 1 egg for 3 polar bodies. Well, how many sperm do men get each time they go through meiosis? Well, each time a male goes through meiosis, that person, or he, gets 4 sperm. Well, that's not fair. Females only get 1 egg, and males get 4 sperm for our 1 egg. Well, in eggs are much larger, okay, in comparison to sperm. Now, speaking of, okay, meiosis, instead of going through 1 round of division, okay, meiosis has to go through 2 rounds. Now, again, you're not getting an exact copy. So say you have 46 stomach cells, okay, stomach chromosomes in your stomach cell. You would get 46 chromosomes for a stomach cell. Well, with meiosis, you only get half, okay? So you're going to create half the amount of genetic information as the parent cell or the regular cell. Now, we just talked about sperm, Okay, and we talked about eggs. Now, look how large this egg is compared to those little. Okay, so meiosis goes through two different divisions, two different rounds, okay, in order to get its 4 end products, either 4 sperm or 1 egg and 3 polar bodies. So, interphase, same thing, G1, S, G2, same exact thing, but now we have 2 divisions. So we have meiosis 1, which involves prophase. Okay, so pretty much looks identical to mitosis, except a couple different exemptions. Okay, so during prophase 1, okay, each chromosome actually has to pair up. Okay, it's got to find its buddy. Okay, its travel buddy, if you will. Okay, we call it homologous. Now, we talked about that prefix homo before, right? Homostasis. What does homo mean? Means same, okay? So again, these chromosomes are from the same trait, but different parents, okay? So again, one from mom, one from dad, okay? So for example, hair color, right? You might get, I have brown hair, okay? My mom has brown hair. My dad has brown hair, okay? So I got my brown hair from my mom, brown hair from my dad. Okay, or a better example would be I have brown eyes. My mom has brown eyes, but my dad has green eyes. So I got my one from my mom, which is brown, and the one from my dad, which is green. Okay, so those are homo. Okay, so with mitosis, there's no pairing. They don't care. They don't need a travel buddy. Okay, but meiosis, they need that travel buddy. Okay, so homologous pairs we refer to as tetra. You might have seen the video game Tetris, right? Tetris means you are using blocks of 4. Okay, so this means that it contains 4 chromatids. Okay, crossing over. Crossing over is a really important event that happens during prophase 1. Okay, this is where those homologous, remember, same chromosomes pair up with each other, but they actually switch part of genetic information, okay? So again, crossing over is extremely important, okay? It's going to produce a different combination that's unique to those individuals, okay? So again, it's going to produce the uniqueness of you, okay? Again, you don't look identical to your parents. You have a variation, okay? So again, that is unique to you. Now, prophase 1, we talked about the homologous chromosomes going to pair up. They switch legs, crossing over. Now, meta 1, what happens? Meta same copies. Now, major difference between prophase and meiosis 2. Okay, so we already talked about Anna. What happens? They're going to separate. And now you have your 2 okay, 2 daughter cells. Now, prophase 2, okay, you're going to go through the same thing. Meta, they're going to line up in the middle. Ana, okay. Which is called N. Okay? So, 1, 2, 3, 4. Haploids. Okay? 4 genetically different cells. We know that. Now, oogenesis means that you're going to produce eggs. So, again, you get 1 haploid, 1 egg, and you get 3 polar bodies. The 3 polar bodies are trash. So, recap. What's the point of meiosis? We're making half the amount of genetic information, okay? So a haploid. Go ahead and click on the link. Now, I've already given this chart to you, okay? So take a minute. I want you to highlight anything that says that it's different in comparison to mitosis, okay? For example, what is the major difference? What kind of chromosomes? Homologous. So if you see homologous, you better highlight it. Okay, so that's the major difference. So again, why do cells divide? A couple different reasons. It's got to grow. It's got to repair because it's damaged. It's got to be able to reproduce. Okay, right? Eggs and sperm. And the cell sometimes gets a little cell membrane. So, I know in math, hopefully, you've talked about surface area to volume ratio. Okay, so it's really important. Okay, so if a cell actually doesn't maintain a certain surface area to volume ratio, again, it's not going to be able to transport anything, okay, through that membrane. It's just going to get too big, and basically it's going to get a buildup where it's so. Let's look at this ostrich egg. Okay, believe it or not, an egg, chicken egg, same thing. It's actually the largest single cell. Okay, well, this ostrich egg is actually the largest. So why is size not limited by surface area? What do you think? Okay. So this is just the recap of. Now, what's number 1? You can start to see those X's. So if you can start to see those X's, okay, it's going from interphase to prophase. Number 2, can't see anything, right? Okay, there's no spaghetti. That's the very start of interphase. Okay, what about letter A? What's A? Normal growth, doing its thing. That's interphase. What about letter B? They're starting to slowly line up in the middle, so it must be meta. What about D? D is where they're slowly starting to get pulled, so that would be closer to Ana, right? Anaphase. Very good. Okay, so number 1, you have 2 brand new cells, brand new daughter cells. So that's telophase. Okay, what about number 3? That would be meta. What about number 6? Ana, good job. All right, let's talk a little bit about cancer. Anybody know anybody that has had cancer or has beat it or passed away from it? Okay. My grandmother has, my dad's mom, actually passed away from, she had a combination. She first had breast cancer, and then she fought it. She beat it, and then she went into remission, and then unfortunately she got leukemia, okay, which is a blood cancer, and she did die. So what happens with a cancer cell, okay, so most cells, okay, for example, if you put them in a petri dish, they are only going to form a single layer. Then they're going to stop. Single layer. Okay? That's normal growth. Okay? It's a single layer. Because they know when conditions get too crowded. Okay? They're going to stop. Say, oh, no, we don't have enough room. We're not going to divide anymore. Okay? So it's kind of like a seating capacity in an elevator, right? Okay? Have you ever gone in an elevator and too many people try to squeeze in on the elevator and you're squished and you feel like you're on top of each other? Okay, so our cells kind of have like a safety switch. They know that we don't want All right, so let's talk about, again, cancer. Okay, so most of our cells have kind of like this safety switch. Okay, they know that if you're single layer, okay, we don't like to be tight. We don't like to, we're going to stop, okay? But cancer cells, they disregard this, okay? Disregard that single layer, okay, because it becomes too crowded. Now, cancer cells will start growing on top of each other, and on top, and on top, and on top, okay? And basically it forms this ball. Okay, or it forms a mass called tumors. Now, there's two different types of tumors. Now, if it's encapsulated by another tissue and it doesn't spread, it's called benign. Now, if it's able to break free and travel towards the rest of the body via the circulatory system, and it's going to other places to divide, it's called malignant. Now, if you go and you get a biopsy, okay, on a body part, and it comes back benign, that means that it's non-cancerous. Now, if it comes back malignant, that means that it is cancerous. Now, what are some treatments for cancer, this uncontrollable growth? The two main common ones are chemotherapy and radiation. Okay, so chemotherapy is where you experience an IV, and we are going to inject some chemicals to kill the cancer cells. Now, radiation is when we actually use wavelengths in order